Today we will discuss about the metastasis in the breast cancer. So how this metastasis in the breast cancer initiate? It is initiate with this calcification in the breast cancer, and that is calcification means the deposition of the calcium on the soft tissues or on the hard tissues, and these calcified granules then go inside the cells, and thereafter it will trigger different signaling pathways such as this MED signaling or different signaling pathways that arises because of the phosphorylation, or we can say it will trigger the phosphorylated pathways such as SMAD pathway, MAPK pathways, and that this MED and MAPK pathways, as we discussed in our previous videos also many times, that they are related to this EMT pathways, that is epithelial to mesochemical transitions. This after this pathway gets initiated, it can trigger phosphorylation of this JNK pathway, ERK pathway, beta T8 pathway, and these pathways along with this MED phosphorylated pathways will trigger EMT, which is epithelial to mesochemical transition. And we have discussed that when the cells acquired the we have discussed previously and many times that when the cells acquire the mesochemicals properties, then it can transit from one part to the another part, or one place to another place. And this is the macroscopic view. What will happen is that it will metastasize, the breast cancer cells will metastasizing from one part to this into this blood vessels, or they are metastasizing through these epithelials uh, by extravasations through these epithelial cells of the blood vessels. And most of these metastasis occurs in the form of bone metastasis, as the calcium is the main part of the bone. So they it is more favorable for this metastasis that is initiated by the calcified granules occurs via this bone. The other things that will also enhance this kind of metastasis is this uh, energy efficient conversion of glucose into the ATPs. And these things can be the normal process is that this glucose is converted into the pyruvate, and then this uh, thereafter converted into the acetyl CoA, and which is the one of the input for this TCA cycles, and that helps in the conversion of this glucose into the ATP. However, when these calcified granules are entered into the cancer cells, then it will trigger the upregulation of this OXPHOS pathway, and this OXPHOS pathway will further enhance the efficient conversion of the ATP, and this will provide the sufficient energy required for these cancer cells, as the cancer cells has the property for the uncontrolled growth rate and proliferations. So uh, they needs excess energy compared to the normal cells, and these things can be supplemented by this upregulation of this OXPHOS pathways that will provide the efficient conversion to this ATP glucose to ATPs. And these things has also been validated, that this OXPHOS pathway has been validated using this rotenone enzyme. The function of this rotenone enzyme is to inhibit this OXPHOS uh, pathways, and thereafter it will reduce the efficient conversion into the ATPs, and this migration of this breast cancer cells will be reduced whenever the cells are treated with this rotenone. So this made the further made the this further made the point that OXPHOS has some important part has an important role towards OXPHOS has an important role uh, towards providing the sufficient energy to the cancer cells that is required for its uncontrolled growth rate or accelerated growth rate, and at the same time this uh, migration of this breast cancer cells from one part from its primary origin to the secondary organ. So I hope you got a basic idea about these metastases in the breast cancers. If you need to understand more about this topic, that is how this metastasis in breast cancer occurs, you can go through the reference in detail. So that's all for this video. Thank you for your time.